Okay, welcome video 4.4 cell cycle. Thinking this one's going to be a bit shorter. We're not going to get through too much in this one. Stages of mitosis will be in the next one. So, starting off, the cell cycle is going to be split into two main phases, which we're then going to split into subphases. But the first one is interphase. This is like the intermission phase. You might think of it, it's like the waiting period before the big main event, which would be mitosis. So this is the phase where cells are growing, copying their chromosomes, and doing all the preparations they need for cell division. We'll talk about the specifics later. And then the M phase is the mitosis phase, and this is where the actual cell division into two genetically identical daughter cells will happen. And I'm going to try and use the phrase two genetically identical daughter cells a lot because that is the output of mitosis, and it's important to just have that drilled into your memory. So, interphase. Like I said, it's the in-between intermission, but it's a pretty bad show because 90% of the cell cycle is spent in interphase. So a majority of its life. And we have three subphases here. The first is the G1 phase, which is the first growth phase. And this is where the cell is going to grow, physically become bigger, make new organelles, kind of essentially become a full-grown adult cell from its baby form when it has just been formed. It doesn't quite have all the machinery in place. And then in S phase, this is the synthesis phase for DNA synthesis. And so this is where we copy our DNA. So going from single pair to, or the single sister chromatid to the double, the X shape. And then finally we have the second growth phase, which is G2. And again, this is kind of just the cell continues to grow and prepare for cell division. It'll do things like making, your centrosomes will form. Your chromosomes will have been fully duplicated, but they're still uncondensed in the nucleus. So it's really just a lot of growing, dividing, preparing, or not dividing, growing, making new organelles, making machinery, doing regular cell life things. So we then head into cell division itself, and there are a few reasons why an organism or a cell might divide, and some of these are very obvious and some becomes less obvious really as you go down. But the main purpose here is for the continuity of life and the reproduction. That's why they divide. It's to continue living and to reproduce. So what do multicellular organisms need cell division for? Well, development still. We still need to be able to develop from a fertilized egg. We also need to grow. When you get physically bigger, you're going to get more cells. Our cells are all about the same size. And then finally, repair. So this could be simple repair from making new skin cells because your other ones are getting worn out. But there's other tissues that will tend to just kind of wear out over time. Like there's portions of your liver that need to be regenerated. Cells in your stomach are exposed to a lot of damage. Generally, things that get exposed to the outside will need to be repaired occasionally. So, three main reasons: reproduction, growth / development, and then actual repair of a tissue. Some notes on how our DNA is organized in this area. We're going to spend a lot of time with DNA in our next couple of units, but quick review, refresher, who knows. All the DNA is called the genome. So your genome is all of your DNA in your cell. Within the genome, within the nucleus, we package our DNA molecules into chromosomes. And then, at least with eukaryotic chromosomes, it's not actually purely DNA. The DNA strands itself are, but they're actually wrapped and wound around these proteins, which we'll get a chance to talk about in the future. So chromatin just means there's protein involved. It's a little more complex than just DNA. When we're preparing for mitosis, though, our chromosomes condense. They go from like a big loose bowl of spaghetti where all the DNA is just unwound everywhere, and it condenses into the actual traditional chromosome shape we think of, which is two sister chromatids joined at kind of the waist, which is called the centromere, where the chromatids are attached. So the two sister chromatids, those are the things that are going to get separated during mitosis and go to the opposite sides to form, each going into a new cell, and they are what will be copied. So right after mitosis, you only have one sister chromatid. In the S phase, you copy to get the second. So, some review. Our cell cycle is 90% interphase, which is cell growth, copying of chromosomes, preparing for cell division. Excuse me. You can think of these as first gap, synthesis, second gap, or first growth, second synthesis, and second growth. Either one works. That's 90% of our cell's life. 10% is spent in the process of mitosis and cytokinesis, which we will get into in our next video. So hopefully that was a quick and painless one. Next one is going to go through the stages of mitosis. See you then. Oh, I almost forgot. I can't forget about that all, folks. Oh my gosh. Bye.